Welcome to week 13 office hour, recapping on the cell replication, over cell replication. Why is cell replication super important to us, first of all? It is important because it will tell us how cells form from, how cells grow, how cells replicate, how cells are used in asexual reproduction via mitosis in eukaryotic cells. It also tells us about cancer biology. It leads us to understand what is happening truly in cancer cells. And it is interesting, not only interesting, but it's a useful information to know about that and use it later on in research for whoever is moving into research. So first of all, what is mitosis? Very basically, before we get into the cell cycle, we had one cell. It's a diploid cell. Diploid, that means has two sets of chromosomes, one from one parent, another set is from the other parent. So it will be a 2N. And the 2N for humans is 46 chromosomes. So 23 came from mother, 23 came from father for the zygote, and later on, one cell, the fertilized egg becomes more, a couple of trillions that make us basically makes up the organism. And this cell is going to go through the cell cycle and forms two identical 2N, 46 chromosomes, two identical sister cells. This is the parent cell, and these are the sister cells. These cells do not have genders. They're called only sister cells, but no genders. These are the product of the one parent cell. I think it's good. Now, let's go and deal with the cell cycle. The cell cycle is a circle. I would just draw it like this. And the cell cycle is divided up into segments. Let's say this will be the G-gap1 segment. When the cell is doing its growth, when the cell is doing its activities. What activities I'm talking about? Making proteins, making hormones, doing its job, maintain its homeostasis. These are the activities what the cell is doing. There will be another, beforehand. From G1, sometimes the cell can go into GO stage. GO means it's not going to be in a cycle. It will not do any division. Some of our cells are the neurons that after certain division, they will stop dividing and no new neurons, nerve cells are going to be produced. Some cells, like the liver cells, they can go back into the cell cycle and do cell replication, but several of them will stay in that stage forever till the end of the life. So the second one is the S phase. This is the DNA replication or synthesis segment, the synthesis or replication. And we have the G to gap2 when the cell is doing its activity and ready for division. Now, all three of them together, from here to there, up till this point, from this point, it's called the interphase. The other phase is the mitotic phase. And the mitotic phase contains the mitosis. Later on, we will learn meiosis, but the meiosis will not go through again and again. It was a little bit different. We will get into later on. Next unit, mitosis. And the mitosis is divided up into prophase, metaphase, anaphase, and telophase. Now, good. Cytokinesis, please recall it. Plants have cell plates formation to divide and make two cells out of the parent cell. The animal cells have a cleavage furrow, kind of like this, and separates the two new cells, the two sister cells. No, these are not sister cells. These are daughter cells. Sorry about that. Daughter cells, not sister cells. The sisters are the chromatids. So daughter cells. What else? Okay, so think about cells, and cells communicate with each other through proteins, and processes that can happen after each other, also regulated by proteins. Now, we have some checkpoints along the cell cycle, and the first checkpoint is at the end of G1 phase. This is going to be a stop point, checkpoint, when the cell has to answer those questions. Answer, basically, if that is going to be fulfilled, the cell can go into another stage in the cell cycle. The questions are, is there any nutrients for the cell in order to grow and do the division? Because for division, has to duplicate its DNA, so it requires nutrients and material to make new cells. So nutrients. Do we have? Does the surroundings have growth factor? Growth factors. Is the cell big enough? How to measure whether the cell is big enough or not? Basically, a surface area over volume ratio. Is that low enough now that the cell will not be able to use the cell membrane anymore as a transport to move waste material out and getting raw materials in, because the cell is getting way too big, so the volume is getting way bigger than the surface area. So these are the questions, and if the cell can answer like yes, 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 well, in this case, the cell will move on this. They pass that stop checkpoint and will start to work on its DNA replication. If one of them is not present, in this case, the cell might not be able to move into the DNA synthesis phase. Like there's no nutrients, the cell normally will not be able to start to produce more cells. DNA synthesis, good. After that, G2 phase, before it starts to get into the mitotic phase, the cell, there will be another checkpoint at the end of G2 phase. when the cell is going to have other questions and before it starts to enter the mitosis or the cell division. For example, is DNA replication finished? If not, well, the cell needs to finish its DNA replication. Is DNA undamaged? If yes, well, the cell can go into the mitotic phase. If the DNA is damaged, no, so it's not undamaged, there's a fixing process, and the cell has to stay in here in G2 phase until the DNA is going to be fixed. The damage in here. The fixing can be, yes, it's fixed, so the cell will be able to go and continue its cycle. If the fixing is not possible for whatever reason, the damage is so big, it's not going to be done. So in here, the cell has to commit apoptosis. Apoptosis, blowing itself up. Enzymes start to get activated, for example, breaking down the lysosomes membrane. The enzymes, the hydrolytic enzymes from the lysosomes, they start to get into the cytoplasm. So breaking down the cell, make the cell a non-living thing because of the DNA, unfixable DNA damage, okay? Now, but sometimes there's no apoptosis. It's not gonna happen for whatever reason. The enzyme that is responsible for opening up the lysosomes membrane is not working for whatever reason. At this point, the cell can bypass the checkpoint and can go into the mitotic phase without fixing its DNA. The DNA codes for proteins, and those proteins are, some of them are regulatory proteins, tells the cell not to move on. But if those regulatory proteins are not functioning correctly, they would not obey the stop signs. They will not stop at the checkpoints if something is wrong. And that's when we are going to talk about the cancer cells. Here is the cell, for example, there's not enough nutrients. The cell will not stop and stay in a G1 phase waiting for nutrients. It will push itself, bypasses the stop sign, the regulatory checkpoint, and will do the DNA synthesis. That's why cancer patients lose excessive amount of weight because the cells do not take into the consideration the cancer cells we're talking about. There's not enough nutrients. They use up the nutrients of the body excessively, not giving nutrients for other cells, and they start to proliferate by themselves. Also, have you ever thought about leukemia? Leukemia is a disease, the cancer of the white blood cells. And leukemia means the people with that cancer, they have way more number of white blood cells than non-cancerous people, not leukemic people. Now, white blood cells are responsible for fighting infections. So we would assume that if somebody who has leukemia probably is the healthiest person because they have tons of white blood cells. Well, that's really not the case. On contrary, people usually go to the doctor like they cannot get better, cannot heal from common cold or from an infection. And that infection lasts for a couple of months now, instead of, a couple of days, they would get better. And this is the time when the doctor orders the blood work and figures out that something is wrong with the white blood cells. Like leukemia will come into the picture. Why is that? So those cells, the white blood cells, are not doing their activities. They are growing, or they are not even big enough to go through the cell cycle, but they just replicate. They pass by the stop sign, the checkpoint, and they just do the uncontrolled cell division. And those cells can do only one thing, is replicate themselves with the damaged DNA. They cannot do activities. They cannot get ready for the division. They cannot communicate correctly with other cells. They disobey. They ignore the checkpoints. Now, one more thing I want to just talk about. Okay, so we are at the mitotic phase. Prophase is when the chromatin starts to coil up and condenses into the chromosomes, the visible ones under the microscope. The metaphase is the chromosomes, the sister chromatids, that is the sister and not the daughter, sister chromatids combine with each other at the centromere with how do we call it? Cohesin protein. Cohesin holds those sister chromatids together. So let's see, we have the chromosomes, and this is the centromere region, and cohesin is a protein that holds these sisters together. Now, I draw a graph in here for you guys. This is the time, and this is going to be the concentration of substances, and we have one protein. It's called the cyclin. It cycles, the amount of the protein cycles along the cell cycle and along the time. And we have another protein, the cyclin-dependent kinase. Kinase adds a phosphate group to a protein to activate it. That is unit 3. Think about unit 4, cell communication. So we have to really just mold these together. And the cyclin-dependent kinase would look like something like this visually. And the cyclin is the circle, small circle, getting bigger, bigger until it's big enough that it will sit here into the cyclin-dependent kinase. And together, they are going to be an active mitotic promoting factor. This is a protein, combine the cyclin-dependent kinase with the cyclin, and together they start to activate the separase enzyme. Separase is a protein, so it's going to get phosphorylized by the mitosis promoting factor, and the separase is going to break down the cohesin that holds the two sister chromatids together. And now at the mitotic phase, this is the last stop, checkpoint, before the separase disintegrates the cohesin, checking, do all the sister chromatids have an attached spindle fiber? Will they be able to move apart from each other evenly when the cohesin will break down? And if that happens at the anaphase, the sister chromatids start to move apart, and at the telophase, the sister chromatids will be at the end, at the two ends of the cell. Nuclear membrane is going to build up around them, and cytokinesis will come in the cell wall form in the plants, or cleavage furrow in the animal cells. Now, if this stop sign checkpoint is not observed by the cells, so the separase gets activated by the cyclin-dependent kinase without the cyclin. It can happen if the cyclin-dependent kinase is coded with a damaged DNA segment. In this case, the separase breaks down the cohesin before the spindle fibers can get attached to each of the sister chromatids, or in meiosis, we will see into the homologous chromosomes, and that's why non-disjunction can happen during cell division. The non-disjunction, we will pick that up in the next unit after the winter break. Okay, I guess that's in nutshell all about the interesting stuff about the cell cycle. I hope it helped you, this video. Have a good one. Bye.